Well, now let's see about certain diagnostic modalities. So we do a tri-screen as well as quad screen for Down syndrome. So in tri-screen we measure the HCG, the nuchal translucency as well as the PAPA. The HCG is increased, the nuchal translucency is also increased. What one needs to remember is that the PAPA is decreased. PA, PPA. What are the other diagnostic modalities? The first one, let's talk about Chorionic Villus Sampling, which is commonly abbreviated as CVS. So this is done at 10 to 13 weeks. So how do we remember that it is done much earlier than the amniocentesis? Maybe we can remember it as the chorion forms much earlier than the amniotic fluid. So therefore the chorionic villus sampling is also done much earlier. Just a clue to, for remembering it. So it is done at about 10 to 13 weeks. So how is it done? So here we have the uterus, and here we have the fetus and the placenta and the chorionic villi. So a needle can be inserted either transabdominally or transcervically, and a sample of the chorionic villi is removed and it is analyzed. So this is how chorionic villus sampling can be done. The chorionic villi will be having the exact chromosomal makeup as what the fetus has. So therefore a sample will be very much accurate for it. So that is the advantage. What is the problem over here? The problem is that it can result in abortion, because a sample of the most important structure has been removed, right? So therefore it can result in abortion. But why do we remove a sample of the chorionic villi? It is because it has the same fetal chromosomal makeup, so therefore it is much more accurate. What about the amniocentesis? Amniocentesis, as the name says, amniotic fluid is being removed, and therefore then the amniotic fluid is checked for chromosomal anomalies. Again, this is more than 95% accurate, and it also causes abortion. We have another diagnostic modality which is known as cell-free DNA. So how is this done? This is also known as non-invasive prenatal testing, that is NIPT. So this is done even before 10 weeks. Here the mother's blood sample is taken. The mother's sample will be containing both the chromosome of the mother as well as the fetus. Once the sample is mother's blood is taken, is collected, the blood is centrifuged and removed, and the mother's chromosomes are removed, and the fetal chromosomes are analyzed for any karyotypic abnormality. It is also very, very accurate, but the problem is that it has a higher risk of false positive. So therefore this can be used as a screening, but we cannot use it as a confirmatory test. So when there is a positivity, then it has to be followed with chorionic villus sampling or else amniocentesis. So this is the problem. Pro is that it is very much accurate. We will be able to diagnose if there is any karyotypic abnormality. What about the cordocentesis? Cordocentesis, as the name says, it is the blood cord is the umbilical cord. The blood is being removed from the umbilical cord. So it is done at about 18 weeks. How do I remember this? So I used to remember like the umbilical cord is in this way. So therefore each is 8 and therefore 18 weeks. So some silly clues to memorize all these. So how is it done? And it is done at 18 weeks. And how is it done? A blood sample from the umbilical cord is being removed and that is analyzed. So but why do we do this? Usually cordocentesis is not recommended for karyotypic analysis. It is mostly done when there is any fetal hematological abnormalities. Therefore we really want to check what is happening in the fetus. Therefore the fetal blood or the cord blood is being collected and analyzed. So this is in short about the diagnostic modalities. I hope this helps.